Welcome to G Fun Facts Online. Today we are going to dismantle a scientific image that has existed for over a century. If I asked you to imagine a virus infection, you would probably picture a single, lonely particle. You imagine a solitary sphere floating through the darkness of the body, bumping into a cell, unlocking a door, and slipping inside. This is known as the lone wolf hypothesis. It is the foundation of almost every mathematical model of disease we have. It assumes that viruses are independent operators, succeeding or failing on their own. But as of late 2025, that image is dead. A groundbreaking discovery from laboratories at Peking University and the Harbin Veterinary Research Institute has fundamentally changed how we understand viral transmission. We now know that viruses are not just solitary assassins. They are cooperative armies that build armored transport vehicles to invade our bodies in massive clusters. Today we are talking about the discovery of the Migrion, a phenomenon that explains why some viruses are so terrifyingly efficient, and how pathogens hijack the very machinery our cells use to move. To understand this, we have to look at what was found. Researchers were studying vesicular stomatitis virus, or VSV, which is a nasty pathogen that attacks the nervous system. They noticed something that physics and standard biology could not explain. The virus was moving too fast. It was spreading through tissue with a lethality that should be possible if it were just drifting from cell to cell. When they looked closer, using high-resolution live cell imaging, they didn't see individual viral particles budding off the infected cells. Instead, they saw the virus hijacking the cell's own movement mechanism. They saw the virus packing itself into massive, fluid-filled sacs called vesicles, effectively building a bus loaded with hundreds of viral genomes. These structures are called Migrions. A migrion is not a virus. It is a vehicle containing a cluster bomb of viruses. While a single virus particle might be 100 nanometers wide, a migrion is massive, ranging up to 3 micrometers. It has a shell made from the host cell's own membrane, which acts as a cloak. Inside, it is packed with viral RNA and proteins. And on the surface, it is studded with viral spikes, turning the entire sac into one giant infectious unit. This discovery is terrifying, but to really understand the brilliance of this evolutionary strategy, we first need to understand the vessel they are stealing. The migrion is actually a corrupted version of a natural cell structure called a migrasome. This is a fascinating piece of biology that was only discovered fairly recently, back in 2014. Think about how a cell moves. When a cell needs to crawl through your tissue, perhaps to heal a wound or hunt down bacteria, it stretches out a foot at the front and pulls itself forward. But the rear of the cell doesn't just snap off cleanly. It leaves behind long, thin tubes of membrane called retraction fibers. These fibers can be very long, trailing behind the moving cell like the tail of a kite. As the cell keeps pulling, tension builds up in these fibers. To relieve that tension, the membrane starts to buckle and swell. It forms these pomegranate-like bulbs along the fiber. These bulbs are the migrasomes. In a healthy body, these are good things. They act like biological breadcrumbs. An immune cell moving toward an infection will leave these migrasomes behind to leave a chemical trail for other immune cells to follow. They're also used as trash bags to dump damaged mitochondria. It is a brilliant system for communication and waste management. But viruses have learned to hack it. In an infected cell, the virus realizes that the cell is preparing to move. The viral replication machinery migrates to the base of those retraction fibers. The virus effectively tricks the cell into thinking that the viral genetic material is just normal cargo meant for export. As those pomegranate-like migrasomes swell up on the retraction fibers, the cell pumps them full of virus. When the cell crawls away, the retraction fiber breaks, and these virus-packed sacs are left behind. They are now migrions. They sit there, waiting. When another cell comes along, perhaps an immune cell trying to clean up the mess, it encounters this massive package. Because the migrion is coated in viral fusion proteins, it doesn't just bump off the target, it fuses with it. And this is where the game changes completely. This brings us to the concept of en bloc transmission. This is the difference between being shot by a sniper and being hit by a shotgun blast. In the old lone wolf model, a single virus enters a cell. It is a race. The virus tries to replicate before the cell's innate immune system detects it. Cells have sensors that act like burglar alarms. If they spot viral RNA, they trigger the interferon response, which shuts down the cell and alerts the neighbors. A single virus often loses this race. It gets chewed up before it can establish a foothold. This is a bottleneck. But a migrion changes the math. When a migrion fuses with the target cell, it doesn't drop one viral genome. It drops 50 or 100 all at once. This massive influx achieves immediate saturation. The cell's burglar alarms are ringing, but it doesn't matter, because there are too many invaders to process. The cell's defenses are overwhelmed instantly. The virus can produce its own defense-suppressing proteins 50 times faster than a single virion could. This allows the infection to bypass the early safety checks that usually keep us healthy. This leads to a fascinating concept called the alley effect. In ecology, the alley effect describes how a population needs a minimum number of individuals to survive. If a pack of wolves is too small, they can't hunt. Viruses are the same. They are stronger together. This is especially true when we look at genetics. RNA viruses, like the flu or coronavirus, mutate incredibly fast. They make mistakes constantly. This means that in any group of viruses, a lot of them are actually broken. They might have a mutation that breaks their polymerase or their shell. If one of these broken, defective viruses infects a cell on its own, the infection fails. It's a dud. But in a Migrion, you might have 45 functional viruses and 5 defective ones. They all enter the cell together. The functional viruses produce the proteins that the defective ones are missing. They share resources. This is called complementation. It means the broken viruses survive. This is crucial for evolution, because a virus that is broken today might carry a mutation that makes it resistant to a drug tomorrow. By traveling in a pack, the virus preserves a massive library of genetic mutations, keeping its options open for the future. The Migrion is essentially a mobile genetic bank. Now let's compare this to other things we know about. We have known for a while that viruses can spread in different ways. We know about exosomes, which are small vesicles secreted by cells. But exosomes are tiny. They are like scooters compared to the heavy transport truck of a Migrion. A Migrion can be 3,000 nanometers wide. It is a heavy hauler. We also know about syncytia, where viruses cause cells to fuse together into a giant blob. But that is static. A giant blob can't move. A Migrion is mobile. And this is the scariest part of the pathology. Because Migrions are formed by migrating cells, they hijack the body's own highways. Imagine an immune cell, a neutrophil, whose job is to rush to an infection, kill bacteria, and then move to the lymph nodes to report back. If that neutrophil gets infected and starts dropping Migrions, it becomes a Trojan horse. It carries the virus deep into tissues that a free-floating virus could never reach. This explains why, in animal models, infections involving Migrions spread systemically much faster than usual. It also explains why these infections are so toxic. When a cell is hit by a Migrion, the massive amount of viral RNA triggers a panic response. The cell screams for help, releasing a storm of inflammatory cytokines. This causes massive collateral damage. The tissue doesn't just get sick, it gets necrotic. It dies rapidly because of the sheer intensity of the initial hit. This discovery forces us to ask, is this unique to VSV? Probably not. Retraction fibers and tetraspanin proteins are fundamental to how mammalian cells work. It is highly likely that other viruses act this way. We know influenza spreads in clusters and filaments. We know polio and rhinovirus can spread in lipid vesicles. It is very possible that what we thought were unique quirks of these viruses are actually variations of this Migrion strategy. Even tumor viruses might be using this. Cancer cells are defined by their ability to migrate and metastasize. If a cancer-causing virus hitches a ride on the migration machinery of a tumor cell, it explains how viral infections can track perfectly with the spread of cancer through the body. So what does this mean for the future of medicine? It means we need to rethink our weapons. Currently, our antivirals target the virus itself. We try to gum up its replication enzymes or block its entry receptors. But we aren't targeting the bus it rides in on. The discovery of the Migrion suggests a new strategy, targeting the retraction fiber. If we can develop drugs that stop the clustering of TSPAN4 proteins, we could mechanically prevent the formation of Migrions. We wouldn't be killing the virus directly. We would be grounding its air force. We could potentially stop the superspread event without harming the host cell's normal functions, assuming we can find a dosage that leaves healthy migration intact. There is also the possibility of setting traps. If viruses love to fuse with these vesicles, could we engineer artificial Migrions? Could we make fake vesicles that look like tasty transport wagons but are actually filled with CRISPR traps or RNA poisons? We could trick the virus into destroying itself. The discovery of the Migrion is a humbling reminder of the complexity of the microcosm. We used to think of the war between humans and viruses as a numbers game. One virus, one cell. But we now see that the enemy has logistics. They have transport divisions. They have cooperative strategies. The lone wolf is dead. We are fighting the wolf pack. And to win this war, we are going to have to learn to dismantle not just the pathogen, but the vehicles they drive. Thank you for listening to this deep dive on G Fun Facts Online. We will see you in the next episode.